we consider a recent study from researchers at Scripps Research, Florida, whose results are already popular in the mass media. They found that the mutation D614G stabilizes a SARS-CoV-2 virus spike protein, which emerges from the viral surface. As a result, the viruses with D614G seem to infect a cell more likely than viruses without the mutation. Many research papers are investigating this matter at the moment. Zhang et al. Observed that the G614 genotype was not detected in February, while found with low frequency in March. Instead, it increased rapidly from April onwards, indicating a transmission advantage over viruses with D614. ViruSurf can be used to illustrate this trend. First, we narrow our search to SARS-CoV-2 complete sequences. Then, we want to limit the collection date to the last day of March 2020. We now have 11,256 sequences. Now we set the condition on amino acid variants. The protein spike and the original amino acid is D, while the alternative one is G. For the position, we set both minimum and maximum value of the range to 614. We then press Apply and obtain 6,592 sequences. The Show Control switch can be used to retrieve, for the population specified with metadata filters, the sequences that do not have the chosen variants. We then find 4,664 sequences without the mutation. Instead, we now want to select the cases of the next period, so we clear the collection date filter and set the minimum to 1st of April this time, by keeping other filters unchanged. There are 23,000 and more sequences collected from the beginning of April 2020 and only 3,331 without mutation. We may be interested in comparing also with results obtained from the GISAID-specific database. We then open the interface from the top bar button. Here, the exact same queries can be performed. To make it quicker, we choose them from the drop-down menu of predefined queries. As there are overlaps between GISAID and GenBank and COG-UK sequences, we press Apply GISAID Specific to perform the search not on the entire database, but only on those sequences that do not overlap with the ViruSurf main database. The same query obtains 15,034 sequences with the mutation up to March, while 80,821 without the mutation. And 18,421 sequences with the mutation after March, against 3,369 after March. If our matches are summed up over the two non-overlapping databases, we obtain that the sequences with the mutation from samples collected up to March represented the 61.6%, while these increased to more than 86% in the following months. Finally, the user may be interested in exploring the table of extracting results. Note that in the last column, the nucleotide sequences can be visualized. If we select from this list the spike protein, we can visualize the related amino acid sequences. Then we can download conveniently the whole table with describing metadata, or just the sequences with the accession IDs of sequences. We hope you enjoyed our example. Please try yourself.